some insights into key concepts, but we will rely on the Globe Netherlands team to introduce you to more in-depth information.
And the other point that we would like to make early is that if you have further questions or if you seek more information or details, we are happy to answer questions, not just during this brief, but also through the email address which we will share with you. We will also share some other resources with any schools who are interested. So there's a variety of resources to support you if you participate in this. And bear in mind that many other teachers have been exactly where you are and have come up with a variety of methods to enact this protocol in a way that is engaging for their students and relevant to the local community and its water.
This is not an official Globe protocol, or at least not yet. Through the work of your colleagues and yourselves, we are gathering data from all over the world, from your experiences, but also students' experiences. As well as the data, the real data that you will collect will form part of a submission to Globe so that this protocol may be examined for its suitability to become an official Globe protocol. So I'm Alessandra. I am from Italy originally, and Sandro is indeed my father, but he's also a Globe Italy country coordinator with Lorella Rigonat, who is not here today. I work at Deakin University. I'm a materials scientist.
And I'm Stuart, and I'm an engineer. I work at Deakin University, but in a different part of the university. And yeah, we have been working on this protocol for a number of years.
We have a lot of interests that bring us together, not just as a family, but with all of you and a lot of students. And through this project, we have noticed that students are particularly in tune with environmental concerns.
One thing that Deakin does for official presentations, and this is an important presentation, is an acknowledgement of country because of the way that our original inhabitants of Australia have been treated in our history so far. There's 60,000 years of continuous civilization within Australia, and the white population, Anglo-Saxon population, has only 230 years. So we do this before each presentation or briefing. We wish to begin by acknowledging the Wadawurrung people of the Kulin Nations, who are the traditional owners of the land on which we here are gathered today. We pay our respects to local people for allowing us to have our gathering on their land and to their elders past, present and emerging. And here's a map that shows roughly where we are in Australia and where our university is located. And this is where, this is outside the front window and door of our house. This is a view from our kitchen. We do have kangaroos in the front of our house.
Well, they're not our pets. They just live nearby, and they're just wild animals, but they are in Australia, I found out.
And welcome to what has become a worldwide initiative. This was prompted by some observations that we had many years ago now, and we tested a protocol very roughly in 2019, and then through the COVID years with a variety of different schools, firstly in Italy, and then more broadly, we have begun to really spread this microplastics draft protocol across a number of countries and through many, many participating students. It's really interesting the different ways that students have been able to embrace this challenge and find solutions to learn about their own waterways. And what we would encourage is the spirit of collaboration, scientific approach, and the rigor of a scientific approach that is consistent, a joint collaborative effort, sharing of ideas and results so that we have quality research, but also continuous improvement. So that's what we're hoping to achieve through this endeavour.
We know that sometimes we are afraid that students will not understand the importance of scientific rigor. However, 3 or 4 years of experimentation have taught us that students love owning the scientific rigor that is necessary to conduct themselves in the laboratory and have clear, accurate, and precise data to present to their community. So we know that while the protocol requires scientific rigor, and we know that there will be mistakes made, it's a learning opportunity for your students and yourselves, but your students will embrace the precision that sometimes is required to produce good data.
And in today's webinar, we hope to introduce this project, its aim, and the phases of the project. We will quickly review the concepts of microplastics. We will look at the context of this protocol and citizen science versus scientific community methods and where this one sits. We will investigate the equipment and the key aspects of this protocol. And there is also one part where we discuss the recognition of microplastics and also the items that you will filter out from your samples, which are not microplastics.
So we have prepared quite a large stack of slides today to show you, but we will go through these quite quickly because we know that the Globe Netherlands team have had workshops with you, with experts, and you'll be able to review all this material in your own time, and you can ask us or the Globe Netherlands team and the local scientists for help to understand the concepts more in depth. So we're talking about microplastics. And in the literature, there are many different definitions. But lately, the one that is accepted is the one by the National Oceanographic and Atmospheric Organization in the US. That states that microplastics are all plastic particles as big as 5 millimeters all the way down to 1 micrometer. 5 millimeters is the upper size, 1 micrometer is the bottom size. But in this slide, you can see that there are 2 vertical lines. These lines indicate the smallest particle that certain methods allow you to find. And what you can see in the first dotted line, 300 microns, is that these are particles that Greenpeace or other large organizations find in the oceans and count in the oceans. However, that is very far from the bottom bit of the range. There are a lot more particles that you will be able to find in that smaller size range towards 1 micron. If you have a good microscope, you will be able to see particles that even Greenpeace does not find.
One thing that is important to understand with microplastics is that there's plastics which are in them. Plastics themselves are mostly made from polymers. This is a key thing. Polymers themselves are many repeating units which might be different from one another, and their structures can often be quite different from the others within the plastic. And so you can have linear, you can have branched, and you can have cross-linked polymers. And it's this ability to control and to mix polymers within a product that allows these plastics to be used in such a broad variety of ways. In fact, they are used by all of us every day. The plastics themselves can become a challenge for us when they're in the environment because the polymers are then combined with additives to become plastics. These additives make the polymers, make the plastics more useful, and they might be flame retardants or dyes, anti-mold, antibacterial, or UV stabilizers. There's a variety of other additives that can be included in a product so that that plastic is optimized for the function it's intended. But within plastics and microplastics, there's 2 broad categories. One is the primary microplastics, and these are really tiny plastics that are micro-sized from the start. And this includes textile fibers, the cosmetics that people use, nappies, pellets, and nurdles, which are often used in a variety of industrial and manufacturing processes. To complement this, there's secondary microplastics, and these are effectively broken pieces of a larger plastic item and make up the majority of the microplastics which are found in the oceans. And if we think about it, most of the plastics that we use are large items, and then a lot of what we use escapes from the recycling loop and can enter the environment in one way or another. And then once it's in the environment, it might become brittle or for whatever reason, break down over time. And so what was originally one large single piece of plastic eventually becomes many, many small pieces of plastics, like we can see here. So mechanical forces, UV degradation, oxidation, and also interestingly, the action of acids, alkalis, and biological members of our environment all combine to break down plastics into much smaller pieces. But the problem that arises from this breaking down of the plastics is that monomers are released, the additives are released, and a number of other degradation products are also released into the environment. And then they can enter the food chain, and a variety of other challenges arise from that. So the secondary microplastics, as they break down, can be a real problem. What is often overlooked is that the microplastics, as well as releasing substances, also become effectively super rafts that can transport naturally occurring species from where they are a natural member of the environment into other parts of the planet. And they can also transport chemical substances around the world. They can also be mistaken for food, for example, plankton, but as a result, they can enter the food chain because they're simply present and animals don't know what they are.
When we say that microplastics act as rafts for microbial communities, we might see it as a very difficult concept to familiarize ourselves with. But if we look at macroscopic objects, we might have all come across a buoy, a rope, or a plastic bottle that has a lot of growth on it. This community of biological organisms has been defined as the plastisphere. And this is a really concerning problem because plastics favor the growth of certain microbial communities where they otherwise wouldn't be. And this causes local imbalance, chemical, biological, pathological imbalance. The plastisphere problem is really, really large because the moment we have microbial communities on an object that is floating in the water, this increases the distance that that microbial community can move. Not just that, everything that is living exudes substances, acids, enzymes, and these contribute to degrading the plastics further and causes the further release of a lot of nano and microplastics. But just to try to give you an understanding that plastics are not just objects that don't do anything, they lose material, they favor the attachment of material, they can cause trouble in the ecosystem in many different ways. The question is, we understand that plastics are a problem and that they are everywhere, but how big is the problem?
One thing that is quite clear is that we don't know how big a problem microplastics actually are, not because people are ignorant, but simply because people haven't been looking necessarily to find the microplastics. But one thing that is clear is that where people have looked, they have found microplastics. And the summary is that from the deepest part of the ocean through to water in the highest mountains, North Pole and South Pole, wherever people have taken a sample and have filtered it for microplastics have been found. And that's common with the many thousands of samples that we have seen, there is yet to be a sample taken by anyone who's participated which lacks microplastics. And really, the true answer is the number of microplastics. How many microplastics or how much that's out there in the environment depends on how they are sampled. So one resource has said that there might be 14 million tonnes of plastic, another one says 8 million, and others have nominated either a number of pieces of microplastic per litre or a total. But the problem is most studies have so far underestimated the number, not through any deliberate oversight, but for a variety of factors.
So the task on hand is to try to help the community understand how many microplastics there are, where are they, in such a way that the data you produce will help your community understand the impacts in the long term. So we set about developing a method that could help with that. And we looked at the methods that are available online, and there's different types of methods. There are very scientific methods that contain a lot of steps, and very community-oriented methods that are very simplified. However, we know that these methods use somewhat rough filtration systems that lose a lot of the particles that might be important to account for.
And one way that we have tried to contextualize this proposed protocol versus existing methods is looking at the question of how many stars are in the sky. And initially, when humans looked at the sky, there would have been only the unaided eye, and people would have been able to count a certain number of stars that were visible and bright enough for the naked eye. And then as technology has improved and we began to use telescopes and other equipment of different power, we could begin to count more and more stars in the sky. There has been, in fact, no real change, no significant change in the number of stars. What has really changed is our ability to perceive and differentiate the number of stars that we can see. And so it is with this protocol.
In fact, the James Webb Telescope has discovered many more galaxies and many more stars than the Hubble Space Telescope had.
So we need to update
So we need to update this slide.
But this hopefully helps explain how this method differs. Now, when we talked before about the different methods and maybe citizen science and other contributors to this field, the question of how many microplastics are in our waters, it depends on how we catch them and the size of our mesh. Certainly here where we live in Australia, there is a very effective community group that walks along the beach and it picks up plastic pieces. These people pick up ropes and buoys and fragments of plastic, such as you can see on the left, but they can only pick up what they can see. So that leaves very many numbers of very small microplastics left on the beach and in the water. If we begin to use a certain size mesh, we can filter out more of these small pieces. And if we use a really fine mesh, we can find even smaller pieces. And these are shown in the photos from left to right here. These are what community members have used so far. And now we have a photo of what NOAA and other groups have used on industrial scale.
So what you're seeing here is a net that is towed behind a vessel, so a boat. It's a net that looks like a funnel. It's 3 to 5 meters in length, so it's very, very long. This is not necessarily the best way to capture microplastics. This net was developed to monitor newtons, which are little organisms that live in the seas. However, in the 1970s, newtons were found along with microplastics, and this method has continued to be used because it's very effective if we're sampling large areas of oceans. However, it has to be effective at filtering large volumes of water, so it has to have large holes. And here's that 300 micron value that we looked at before. Everything smaller than that just washes through. Further, it's not useful for schools. Schools and communities might have access to sieves, as you see in this picture, and other methods that are more accessible, but are often more directed towards educating the community than gathering data of importance to a scientific effort, which means these are typically inaccurate, and there are lots of different efforts all over the world that we cannot put together because the method is so different.
And this is where we hope to fill a gap that exists between industrial or commercial scale sampling and community versus maybe the reality. And this method has been designed and proposed to be scientifically accurate, but also accessible for people of different ages and abilities. In other words, for community members and school groups who might be guided by experienced and capable teachers. And so we have found that the participants are able to identify even small particles from 5 millimeter all the way down to our other end of the scale. And it's a much greater degree of accuracy and consistency than in many of the scientific approaches. These are some of the photos from some of the schools. We have high school and primary school aged participants from several European groups. This method has been tested and trialled and validated by teachers and students from technical schools, as well as from more classical schools and high school, all the way down to very young students, like you can see on the right. And each participating school and teacher has been able to provide feedback to facilitate continuous improvement of the method. And what we have now is the result of quite a lot of practical application, refinement, and review.
But to answer a question you might be asking, is now important. So we're talking about a protocol, but what are we looking for? We wanted to clarify with you that when we talk about microplastics, we consider among these plastic pieces, so plastic particles, all those particles you and I would consider as plastic, but also all textile fibers. And this is in line with the most recent advice to the US government by the National Oceanographic and Atmospheric Organization. And when we talk about textile fibers, we talk about what we're wearing today. They could be cellulose-based textile fibers, like cotton or linen, animal-based textile fibers, wool, cashmere, typically animal hair, and man-made textile fibers. And these could be synthetic, like polyester, acrylic nylon, or regenerated cellulose fibers like bamboo, for instance. These are important to mention because they are all fibers that are found in the environment, and their impact is so far difficult to measure. So what you're doing is something that is ahead of much of the scientific community. The protocol is reasonably simple. It involves sampling a sample of surface water, like you do in all GLOBE protocols, and use techniques and tools that are reasonably common in schools. We will support you through reference materials that the GLOBE Netherlands team has kindly translated for you, doing a fantastic job because it's all in English otherwise. The protocol consists of a series of instructions, really, like a typical GLOBE protocol. But in the files that you should have a teacher's guide as well that provides you with key concepts or frequently asked questions to guide you and your students through the protocol. These instructions are for different settings. They could be field guides or laboratory guides, depending on what step you want to do and where you want to do it. The tools are pretty much like this. They consist of sampling systems, like in these water bottles on the right, filter membranes that look like flat discs, filtration systems like the yellow tower looking like system. And as you can see, students use microscopes and they can use them even outdoors in a lot of settings, especially during COVID. Protocol steps are very simple. Like in any globe protocol, you would collect the sample in a clean bottle and take note of the conditions and when you sampled, as well as temperature. And then you would take the sample and filter it on a microbiology filtering unit. Once you have filtered it, you can put that filter away and look at it in a couple of days or observe the sample immediately. What we are doing on the filter is capture everything that we can capture on a microbiology filter. You'll have to be a little bit careful because you will see a lot of life under the microscope that you have captured through the filter, but sand as well. So what we're going to try to do today is show you a couple of examples of what you can come across with your students, because there's a completely different world in the water.
And what you can see here is several photos of different ways that participants have been sampling and different locations. Some have incorporated this into field trips and other activities, but the important thing is that these photos show that a very broad variety of people and settings can be used for sampling. And what this allows participating schools and teachers to do is to identify a body of water which is important to the local community, and then to undertake sampling there in an accessible location. That then means that the student and the community will really feel some value from the results that we derive from the samples. When doing this sampling itself, the standard recommendations are used to use a clean bottle, to wear gloves, remain downwind compared to the sampling site, rinse the bottle, and then cap it. And we talk about doing 3 samples in each location, 3 different sampling bottles from each sampling site. And also to cap the sample immediately that you have taken that sample. And some schools have stored the sample because there might be a week or so between undertaking the sampling activity and then the filtering. And if storing any samples, the recommendation is to use a fridge or a freezer for that. Some schools have had to reach quite a long distance to safely take a sample of water, and some schools have built their own sampling rod, and we have some examples of that within the reference material. And here we now move on to the filtering. As you can see here, students and participants have been able to undertake the filtering process both within the laboratory and outside, and interestingly have come up with different solutions based on the equipment that they have available to them in the school. My favourite photo from all of the participants so far is this one here, where one school could not find a way to create a vacuum in the lower half of the sampling unit. And so one student borrowed his family's vacuum cleaner and brought it into the school to create the vacuum and speed up the process. I applaud their inventiveness, but I hope that their parents were forgiving when they returned home. But most schools have been able to use another method, which might involve a syringe and some valves. And again, we have some recommendations about how to acquire and use that sort of method instead. This picture here shows a Nalgene reusable filter unit, which is one of the units that many schools have used. It doesn't have to be a Nalgene, but this is the most common unit available at the moment. If your school has them, then these are very, very useful. So long as the school is able to acquire something that is able to fulfill the function that this unit does, then participation will be really quite simple. At this point, we'll talk about some key points to remember. When storing and sampling, all of the equipment should be stored clean, and we recommend that the students and the participants use air drying only and not wipe down any equipment with paper towel or cloths. That might leave fibers within them. And when sampling in particular, rinse all equipment that you use with deionized water immediately before using it. Rinse the sampling bottle with water from the body of water that you're sampling downstream and downwind from where you sample. Take 3 500 ml samples for each site and each sampling event. And then we encourage people to sample at least 2 occasions during the program, and also to take photos of the site as well. These are always very interesting for people to see.
And it's documenting your activities, your school activities, and you can showcase them with your own council or even within Globe. We know a lot of students that have presented for IVSS, for instance, or other science fairs. So they have turned into very, very useful documents.
And some more points to remember. Laboratory blanks. Use deionized water for a laboratory blank so that students can learn and understand the process before using sample filters or other equipment. It's really important that students are confident and competent in the various stages before actually undertaking the sampling. And with the filtration, if the sample is really black, dirty and murky, the advice and recommendation is to filter that sample across several membranes. For example, the 1 500 ml sample bottle might be divided into 200 mils, 200 mils and 100 mils for each of 3 membranes, and simply record the volume of sample that has been filtered by each membrane. And then once you've checked them all, you can add the values and you know how many microplastics were within that 500 mil.
One way to ensure that your sample is not too opaque is to ensure you're not picking up sediment. That can be difficult for certain students and for certain water bodies. However, if you can avoid to collect sand, it will pay off.
And always rinse the membrane with deionized water after filtering your sample to remove any salts. And remember to put the air filter on top of the filtration unit lid. There's been a number of groups who almost forgot it on occasions.
The other reason for having laboratory blanks is to understand the level of background contamination that you have where you are working. Microplastics are everywhere, and it's important to actually measure that. If you're running a laboratory blank, if you find microplastics on your filter, they will not come from any sample. They will just come from the air and water around you.
That was demonstrated practically by one of the participating schools early in the process, where the teacher and the students found a whole heap of mica or something in their samples, and it turned out it was from either the teacher or the students were wearing makeup. And so from that point on, the teacher said, no makeup for any participants, be very careful. And that was a really useful contribution to learn that the background noise within the laboratory would normally include these contaminants.
So these are all slides we will share with you, so you can go through these in detail in your own time. But really, it's common sense. If you stop and think, and even look at the dust that is settling on your wardrobes and put that under the microscope, that could be a useful exercise for your students to understand that microplastics are actually everywhere. So the last bit of the work for you would be to then observe and analyze your samples. This is where you can have 2 reactions in students. One reaction of disappointment if they do not find any or many microplastics, which is contradictory for an environmental education class. The second reaction is probably connected to the first, fear of not finding anything, And students will tend to identify everything that they find under the microscope as microplastics, including plankton, algae, pieces of animals, or pieces of plants. So just something to be aware of. Students are very enthusiastic, but they have very little awareness of how small things are under the microscope and how natural things might look like under the microscope. It's going to be a long journey for your students, potentially, if they have never seen a microscope, but they're really good at learning. However, it's important that students familiarize themselves with what reference materials might look like. Put a t-shirt under the microscope. Let's have a look at textile fibers. What do they look like? Also use the microplastics recognition guide that has been translated by the Globe Netherlands team. And don't be rushed, because it's okay to take time. We have mechanisms in place for us to check your data, and we will show you how that works. In order to check data in particular, you can use your mobile phones or your students' mobile phones to take photos through the eyepieces of the microscope if you don't have a computer. We know students have great hands at doing this. This is what a filter membrane looks like after filtering a freshwater body. What you can see in this picture is the membrane that is clean, that was just held down by the filtering system, and the area that has filtered all the solids present in the water. We have some arrows on the membrane and some letters. We invite students to write letters on the membrane, and we have a couple of videos to show you how to do that before they filter. This helps them not become lost when looking at a filter. And we invite you to follow the lines in the membrane to ensure that you're scanning every single square. There's a couple of ways to do it, and one way is to take coordinates. 3 rows up, 6 columns to the right. This will become a 3 north, 6 east point for something that I observe. And this helps your students be precise in identifying their materials, taking a photo, and recording where that photo was taken. You can do this in a number of ways. You can even get students to take photos, but also draw what they see on an enlarged photocopy of the membrane that you have. The last bit that is important is the calibration. How big are things under the microscope? What do things look like? How do I measure under the microscope? And you have a variety of ways to do it, but the simplest way we have found is to use the printing that is on the membrane, that square grid that you saw on the membrane, for reference. Some students took a photo with a mobile phone and then enlarged it, and just using proportions and a ruler, like a school ruler, were able to quite accurately work out how thick that black line that is the printed membrane was. And from there, they could roughly measure the length of a fiber. Some membranes have dots, some membranes have lines. However, work with your students and put this to them as a problem. We don't have a ruler inside the microscope. What becomes our ruler in the microscope? There are some suggestions here that you can follow. Again, they will be in the slides that we share with you, but you might have better ways to do this. So we invite you to share that with us.
One thing that we have been able to develop over time is the microplastics recognition guide. This is a comprehensive document and has been contributed to by a variety of participants so far. In this summary here, we show there is some text in the guide. There are some indicative or representative photographs of different types of object that have been observed under microscopes. And then there is a little a process that you or your students can follow to ask yourself, or even as a group to ask one another how the fibre, how the object might be classified. So we can go through those in more detail, but these are all also included in the reference material, which we can share with you. These have been very useful for particularly the younger students to help them be more involved and more accurate in their classification. One thing that we'd like to point out is that when we look at these items under the microscope, it is the world at a different scale. And while we might in everyday life observe objects and items in a particular way and interpret them and classify them using our eyes and what we consider normal, under the microscope, the world can appear different. So biological debris is something that looks quite dusty and not well identifiable. Microorganisms might be round or lenticular. They could be micro and macro invertebrates or plankton. There could be filiform objects and just weird objects. We've got photos of everything and anything that might be found. There's biosilica and shell type residue, and there's also quite a lot of animal hair, particularly in rivers and lakes and streams. For the biological debris, we recommend that you look for natural colours, brown or green, which are warm colours or transparent, cellular structures and regular structures on this very small scale, and some which are clearly biological in shape. With fibres of biological origin, these do not need to be reported, but they're often green or brown in colour. They could look like a bit of a scribble or a very uneven line. They might have branching coming off them and be of segmented structure. There might be very straight items and many, many of them in the water.
I'm just going to say, these are not cotton fibers. These are not textile fibers. These are just filaments that you can find in the water or things that look like
algal
fibers.
colonies or something like that. So there's a great number of different examples, and we have many photos available to help. But here are some of the common encounters that people have had. In this example, this is a soft and irregular filament, a biological shape. This could have been the roots from an aquatic plant. We have here some more examples. We're not sure what this one is in the middle, but these are all biological filaments and don't need to be reported. And then in this very interesting image, we have a cellular structure here in green, and then very many straight examples. These needles are plankton, and this is probably an algal structure coming through here.
Yeah, so filament algal colony. But everything else in this picture is basically biological. This is life. This is plankton in a freshwater system. And so
it can be a very busy image and a very busy world that the students observe, but not everything will be a microplastic. Other debris that does not need to be reported, often green or brown, dusty. They might look like sand, round, regular, and many, and biological shapes. And interestingly, at this scale, this is a naturally occurring organism. If we were to look at this in our normal vision, we would assume that this was something that humans had produced through some sort of manufacturing process or maybe a video game or something like that. But in the micro scale, this is quite a fantastic example of biology, of life. And here we have an algal colony as well. With sand looks like shiny debris. It might be flaked. There can be crystals and hard particles. So we look for very shiny surfaces with sharp edges and earthy colours.
We're pointing these out because often students see something under the microscope and I think that's a microplastic. Actually, no, that's a grain of sand, but it does look very different from what a grain of sand looks like in daily life, which is dull, not shiny.
And that's because of the different ways that light can diffract, refract, and reflect when we look at it at this scale compared to with our unaided eyes. Now we move on to microplastics at this point, and there's the textile fibers, which might be cellulose, such as cotton, linen, animal, which might be wool, cashmere, and similar, and then manufactured or man-made polyester, acrylic, nylon, and the clothes that I'm wearing. There's plastic particles. These can be identified by their color and their surface appearance. And then there is always the possibility that there's unknown items, which could be plastic, but which we are unsure.
So we're just going to point out this report by NOAA that we mentioned earlier. This has been submitted to the US Congress, and it was submitted in October last year. And this is simply the exponential progression of the number of publications that are reported in terms of microplastics and microfibers in this case. This report indicates the importance of cataloging every type of microfiber we come across. So among them, these are the most common you'll find around in the water. Textile cellulose fibers, we can easily call them cotton because that's the most common plant-based fiber that we can come across in textiles. They are often mistaken for man-made fibers just because they look transparent under the microscope. And we're not familiar with a white cotton t-shirt being fully transparent. However, that is what things look like under the microscope. In the macro scale, things look opaque. In the microscope, actually, our t-shirt is a see-through t-shirt. So here is an example of two cotton fibers, which you can tell are of textile origin because they are blue. We'll give you some hints about how to recognize that they're cotton and not man-made. They oftentimes look like a paper ribbon. They look a little bit uneven, squashed, paper-like rigid structures that are bent and twisted. And these are some examples from some colleagues of yours. These are photos taken by students using a cell phone through the eyepiece of a microscope. And you can tell that there are similarities in the vague shape. However, cellulose fibers, but textile fibers are great distractors. They take your attention away from other things that might be in the sample, like that little blue particle in the center of that red circle. It's important that your students pay attention to everything they see, not just the big things. These fibers are really large. There's also smaller particles in there. You will not come across many textile animal fibers. We're talking wool more than anything else. But if you do, this is what they might look like from a top-down reflection microscope. You can see they have a cylindrical section, much more regular, much straighter, shall we say, than the cellulose fibers that looked all twisted. And sometimes you will see reflections that indicate that they have scales like our own hair. There's another couple of examples here, and you can barely see the scales on here as reflections change. And then there's man-made textile fibers. These, again, could be bamboo fibers as well as synthetic fibers that you see listed here. The section is a bit different. They often have a channeled structure. So their section could be round, it could be square, or it could have a channel inside. And these are quite obvious to recognize, but there are not many in surface waters. There's often quite a number in sediment. This is an example of one man-made fiber together with a few bits of biology in there. couple more examples here, but we have a bunch of images that you can freely browse that have been shared with Globe Netherlands. Other microplastics you might come across are going to be colorful. You are most likely going to see red, orange, and bright colors under the microscope, and you will not maybe see the white or non-colored particles, and that's okay. Just take photos of what you find. We find that blue is a very common color, as well as red. And so we keep finding bits that obviously are not biology, surrounded by biology. And this is something your students will see. And if you look at the scale of these pictures, you notice that these are much smaller than the 300 micron particles that NOAA report all the time. But then what to do once you've taken the pictures and made comments about the particles you have collected? You fill in a data sheet, and you have an example with the Globe Netherlands who has translated this for you. We ask you to fill this in carefully and select various aspects of the data sheet and pick the best guess. But also try to identify what you think something might be. For instance, this student has identified this as a cellulose textile fiber, and they have put a link to the picture. So you as a teacher can open that picture, evaluate that, yep, this is a textile cellulose fiber, and validate the data this way. By doing this, you provide us and Globe with a large data set of images, but also of data that we can validate. And that's really important from a data quality perspective. This is basically most of it. We have a couple of examples of equipment, if there is time, or if there are questions that are related to that. But other than that, we're here to answer your questions or comments or anything else you might want to hear from us.
Ale, the counter of the data sheet.
That's a good point. Sandra is reminding me about the counter. So when we have a data sheet, you will see that you will select here your observation as well. So in this case, we had the cellulose textile fiber. This data sheet has an automatic counter that will count how many cellulose and animal textile fibers, in this case, how many man-made fibers, plastic pieces, and unknowns, or things that you're not sure about, have been identified in that sample. If you take these values and the volume that you have filtered for that sample, you'll be able to measure or calculate the concentration of each type of object per unit of volume. In general, teachers and students have found 10,000 particles per cubic meter up to 250,000 particles per cubic meter. And most of these particles are plastic pieces and cellulose textile fibers. These values are much larger than what you oftentimes find in the scientific literature. And it comes from the fact that you're using a very, very fine filter, while in the literature they use these very large filters. So this counter is really important. We suggest that you use one data sheet for 500 milliliters. And so if you have 3 membranes, you put membrane 1 up to here, membrane 2 from here to here, and membrane 3 to follow. So that the same data sheet has the total of the observations for that one 500 ml bottle. You can duplicate this and do the second bottle, etc. Was that helpful?
For me, it was. So are there some questions from the teachers? It's quite a lot of information, I guess. Most of them didn't start it. They all got this filtration unit from Globe the Netherlands with a couple of membranes. Are there some teachers planning to start on, let's say, next week already? This week or next week?
What do you think you'd like to do before you start?
Well, I know what I would like to start with, because one of the things I think my students will have lots of problems with is identifying what they see. And what you just showed me makes me wonder whether I am able to determine myself whether it's from biological origin or from man-made origin. Because I'm a chemist, so I'm not a biologist, and I have not a lot of experience in seeing these micro world things. So I think that the first thing I will do is for our next, we always have at the end of the year this meeting with teachers. I think this is a very nice way to do something practical on that meeting. Then and I will ask my colleagues to bring a sample and to determine the samples ourselves. So next school year we can start to let students observe.
I'll bring up a picture while we answer a question online, and then we're going to answer a question on the chat, but you have raised a great point. I'm sorry, we missed your name.
I'm Jeanette Brouwer.
I'm sorry, I'm not going to be able to say that.
Janet. Janet.
Janet.
Or Janet. You can say Janet.
Janet.
But one thing, while Alessandra brings up the material, one thing that we would point out is that there's been teachers who are of scientific background, such as yourself, but not in biology or not used to classifying in the microscale. And there's also been teachers participate who are not necessarily technically minded. Many people have lacked confidence at the beginning, but that is why we have, with their help and with their guidance, developed a guide that can help with learning how to classify the observations, the items that you observe. Also, there is quite a comprehensive image database, which has a very broad cross-section and deep number of examples of each type of fibre. And we're quite confident, based on the evidence that we've gathered so far, that the waters that you sample will probably produce similar results to what has been reported elsewhere in the world. There's some small variations unique to some places with particular industries, and that's where it is really interesting. But confidence is something that does come with reading the documentation and reviewing the photos and just trying it. And then we are always happy to help answer questions if you have some.
So, Janet, I'm just going to ask you, this is a control sample, right? This is a sample that a set of students prepared to train themselves. What did this sample come from, do you think?
How do you mean? Where did this sample come from?
The students prepared a sample of known composition. Mm-hmm. Water that had fibers in it, and they knew what fibers were in the water. How do you think they prepared this sample? This sample was prepared from the washing machine. This
Well, that was what I was thinking already, that I
Yeah.
would take a sample of the washing machine. Okay,
Perfect.
yeah, or make my own sample from the fibers from the you call it a dryer, I think.
The dryer,
So-
perfect.
So what I find really fascinating is that you're already thinking along the same lines that some earlier participants have. So this gives me confidence that you're heading in the right direction.
Yeah.
Well, I am very confident that we are going to learn how to identify it, but it's just that I, for myself, I would like to see it before my students see it, just because I'm so curious about what I'm going to see.
Yeah, absolutely. Yeah.
And so are we. We are really interested to see what your students uncover, because they will probably be the first students to sample this particular water body, and they'll become the experts in the microplastics that are found in there.
And if you prepare reference material with samples from your dryer, from your washing machine, from your students' washing machine, then you can print them and keep them in the classroom and work out what looks like that. And so have that reference scale that belongs to you. So don't just trust ours, do your own.
Ale, there is a Rob question, answer, a suggestion by Rob in the chat.
Rob is saying, in order to measure the length of the plastics, he uses a digital solution, a software called MyCam, and you have a 5 megapixel camera on top of the microscope, and you can calibrate it with a micrometer glass. Rob, are you at a university or at school?
Okay, at a school. That's a fantastic level of equipment for a school. What we have found is that some schools have partnered with a nearby research institute or university to gain access to not only the hardware, the microscope or the other equipment, but also to the technical competence from the scientists and technicians who make use of this equipment. And the feedback, incidentally, the feedback from the students who've been able to see science in its applied form used by professionals for their job has actually helped inform some students about what they might want to do in future. You know, science can actually make a contribution. So that's another benefit. But even if you don't have 5 megapixel cameras or high-tech microscopes, there are various methods to gain access to them.
I think, Rob, it would be fantastic if you could share the information with Globe Netherlands for broadcasting to other teachers who might be interested.
Fantastic.
Thank you.
Thank you.
Rob, can we see you? Otherwise, you could be an artificial intelligence.
No problem, Rob. No camera. we understand.
Yes, so Rob, if you can tell me more indeed about the camera and the software, then I can add it to the teacher's material. That would be really nice.
And Rob's suggestion has built on some of the feedback from the students. The scary thing is that students in many countries have their own very high-tech mobile phones with really high fidelity cameras, and they're very used to taking photographs for all the social media things that they do. And so they can take some very high quality images, just holding it above the microscope's objective and taking the pictures far better than I could ever hope to do.
Can I ask one more question?
Of course.
Janet here, because I'm working for a primary school as well, and at first I wasn't thinking of letting these children look at the filters, but do you have, are there already a lot of primary schools taking part of this?
Yeah, excellent question. We actually have put the Globe Netherlands team in contact with a researcher from University of California, Berkeley, who has been working with colleagues from San Francisco to develop a teacher pacing guide with the I guess, educational content that is available to primary school students, as well as middle school and upper high school students. So they have developed a series of activities, and there's a video. Dorian, maybe you can share that video that we shared with you by Tracy. And maybe Tracy, it would be great if Tracy could share with you that teacher pacing guide that she and Matt Ferner developed. And if you could share with Janet, that would be great. But
in addition to the American schools, there's been Irish primary school students participate, and very young students from other countries. And they're actually, the smaller students fit into smaller locations, so they can often get samples from water bodies that bigger students won't get.
Yeah, or you can adapt it to the schoolyard or the dust that we were talking about, and putting, finding out where the dust is in your bedroom could always be interesting for students.
Mm-hmm. Okay, I'll get in touch with you, Doreen, about this.
We've had a question, where can we buy new filters? The second part, can we reuse them? No, we can't use them. For one thing, they're really, really fragile after they've had water pass through them. And so it's very easy for them to be damaged if they're handled too much. They wouldn't be able to cope with cleaning and being prepared for reuse.
This is a brand we have been using, Alstrom, as well, but there's Millipore, Merck. Some of your colleagues ordered them from AliExpress, which is great. So long as they have a printed grid, they should be okay.
And the participants who've made use of the filter paper that has the grid, whether it's a solid line, dots, waves, or something else, it really does help integrate this protocol trial into, for example, mathematics or proportions or whatever else. You can, again, give a really practical and tangible demonstration of how maths can be applied to help solve real-world problems.
Rob is clearly in a really good school. You have an infrared spectrometer that they use for forensic experiments, identifying pieces of clothing. Yes, Rob, that's a great point, and we invite you to use that, especially on pieces that you can see. Many of the particles you will collect are smaller than 100 microns. So it will be very difficult to pick those out and put them on the FTIR, which is the infrared spectrometer. However, if you find bigger pieces, absolutely. That would really add to your students' experience, but also pieces from the backyard, a broken pot, a plant pot, or rubbish would be great for the students, I think.
It might actually be something where you could look at, say, some of the larger pieces of something. If you have an object like this pen, if it breaks, or if you find something that is broken, you could look at the pieces of different size, for example, and then also explore secondary microplastics and how they can be developed so quickly.
And even the top of a book, the cover of a book, I guess this is a postcard, they have plasticized coatings, so they have plastic type coatings. Even discovering things like these could be important for your students. So realizing that there's plastic even when you don't think there is plastic. The cover of this book is shiny. This is a plastic coating. What is it? That would be really helpful, I think. The other thing we can highlight is that a lot of students became detectives in their own communities. In the community of Mantova, which is where Sandro is, they found a lot of red microplastics in the 30 micron size. And they went upstream and downstream from a certain point, and they could pinpoint where the particles were coming from. And they probably come from boating. There's a lot of boats, and they either come from sandpaper used to remove old paint from the boats, or from buoys that are in the water and very old. We have a comment, Dorian, would you like to share? There's a science fair.
Yeah, thanks. I was just wanted to tell the teachers that if you are planning on starting the microplastics within the next week, then you should come to the science fair. We organize the science fair, yearly science fair. And it's very nice that the embassy, the US embassy, they give us the opportunity to have 3 prizes for the best teams who did a presentation in microplastics, and they get this behind the scene at the seal, how do you call it? Where they get the seals from the sea and get them back to the sea.
Sanctuary.
Yes, thanks.
Sanctuary.
And they also do a lot of research about the microplastics and plastics in the sea. So the 3 best teams, they have the possibility to get behind the scene and do a plastic cleaning with the boat or on the side on the water. So that's really nice, I think. So that was just information about that.
That's awesome.
I see also a Dutch question. How can I share the information with Globe about the software and the camera? Okay, Rob, you can just email me, please. I will put my email address in the chat. Yes, so you can just send me an email. Thank you so much, Rob.
Have we scared any of you? Is any of you scared?
Because we can we can try harder. If you're not scared, we can try it again.
I would like to remember that there are interesting learning activities, like to measure the density of the different plastics. So it's a very interesting learning activity that you could manage at school with your students, collecting different kinds of plastics in your house, on your student's house, and measuring the density or just seeing if they float on sink. It's very interesting because students don't think about the density, but density is a critical aspect, critical parameters concerning the distribution of microplastics in air or in water on soil.
We removed a lot of slides because we typically deliver this over 2 90-minute webinars to spread the learning a little bit. So tonight we removed a lot of slides. I know you saw a lot, but in the interest of time, we removed a few slides talking about special activities that you can undertake. But yes, learning about the density of things, the size of things, the concepts of surface area in things, and how smaller things and flatter things travel further is really important. That's an excellent question, Rob.
It's when students are in a group, if we support them and give them the information in a logical sequence, has been a really useful way to keep students interested, but also awake. It's very easy for this to become a very technical and detailed subject, but interestingly, it is also something where there's a lot of opportunities to integrate this into different parts of the curriculum. For example, some students have, or some schools have used this with politics or with social science or with mathematics, physics, chemistry, etc. And that way you can really stress certain parts or integrate it into what you're teaching. But basically, the key thing is share the resources that we share with you and show them that there is a key that the student can follow. And then something simple like asking the students to draw by hand what they observe under the microscope, then they can share that image with other people, and drawing it by hand, they convey the key elements of what they see, and then that is how people can classify it.
The other aspect to keep in mind is that students are curious, so they will seek information and seek to judge themselves whether the information is good, bad, useful, or not useful. The important aspect is to make them feel in control. We know that students can select the water bodies they can sample, and they feel in control of that process, but also they can be made responsible of who they share their data with and what they share and how they present it to the community, also making them responsible for not scaring the community. It's not that microplastics are there all of a sudden because we're finding them. They were there before, we just did not see them. And the important thing is to measure the level of microplastics now, so that if we have any intervention, we can see if things are improving or not improving or getting worse. So I think empowering the students to make choices as if they were scientists, giving them information without judgment is key to their positive involvement.
And there are very many resources which we can share with participating teachers and schools. And these, if teachers familiarise themselves with it first, but then can share the key elements with their students, then everyone is learning together, the whole class is learning together, and the confidence will be there.
Do you know what is our dream? That you are able to build a database about the microplastic in the Netherlands, a database covering all the water bodies in the Netherlands. But this is a dream that is covering all the world. So we are thinking about the big communities of students and teachers telling their community, telling their territory, what is the situation about microplastics, in order to grow the awareness about this big problem, this emergent problem.
And in actual fact, we had one case study where one class was able to do some microplastics analyses, and then when they sampled this particular water body, it allowed the local authorities to work out that people were dumping rubbish upstream from this water body, and I think it was the water supply.
It was in a cave. That had the water table. So the students sampled the water from the water supply for the town, and they found microplastics, which indicated there was a source of rubbish. Hello, Maike.
Hi, Maike. Very nice to meet
Hi.
you.
Nice to meet you again.
Yeah, very nice, Mikey. You're here. We just got some questions of a teacher. He said, well, how can we get this hard in recognition, all the particles? How can we help the teachers? How can we help as a teacher to help the student to get them to do it the correct way? And I thought maybe you can tell something about how your student did it. How did you manage as a teacher to do it right?
Yes. One moment. I changed my space. because I'm in a lesson on this moment. So there's a lot of walking around me. How do I do it? I take the microscope with the students and I let them check what they see and let them make a video of it. And then I see the video and then analyze together with the students the video of the microscope.
Because it was still hard for the students to do it just by themselves.
No, not all, but for some students it was a long time ago, because only in the first class of biology they take the microscope. In the other class, like second class and third class, in my school they have no practical with the microscope, so it was a long time ago. That's the reason that I do it together with them, because for some students it's difficult, but when they can see the parts and they see a smooth part of a roof part with it, then they recognize the difference between the particles.
And what did you help Maaike to get to know about all these microplastic structures or biology structures?
Look a lot of time at the microscope. So first I do it by myself and check all the parts, and all the time what I see, I compare it with particles of globe in the pictures they make. So all the time I compare it, and also I check on the internet, and that after that now I can see it and I recognize the particles. And also the students make a lot of pictures, and also they want to know it. So when they send me something and I don't know what it is, I will check it for them in the evening or later on the day.
Okay, thanks. And was the recognition guide also helpful? Because you have also a lot of
a lot
by words and pictures information about different particles.
The pictures are very easy for students to compare what they say in the recognition guides. It helps a lot for the students. And also they have make a card that is it yes or no, and that part, that's the first step I do. Yes, the searching card.
Great. Does anybody has a specific question for Maaike now during her lesson? No.
Any suggestion, Maaike, you might have as to how to enable students to own their project? Because your students were particularly proud of owning what they were doing.
Yes, and still they are still doing it again. Every year they want to do it again to see what's the difference about the surface water. So that's very nice. I've now students, they will want to do exam in this typical subject. So that's very nice. What helps was the first time they checked the water under the microscope, and they see so many particles. They were very shocked, and after the first shock, they want to see more and more and more and more. And it's not good for the water, but it helps to feel connected with the project. Because I think we have a lot in the water in Netherlands. You have seen the results. Do we have more in the Netherlands than other spaces on the world?
It really depends, Maaike. You've done a lot of work and your students have done a lot of work. So we were trusting their data, and there's a lot more resolution where you are than in other areas. Mantova is head to head with you guys.
Okay.
Unfortunately.
Have a good stats there. but yeah, I think you're right.
Yes, and I let them free. So I don't give them a place to do the research, but I told my students, where do you want to do the research? It could be everywhere in the Netherlands. So some students travel to the south, or some students travel to the north, because it's their project and their research, and that helps a lot to get involved of the project.
And I've got a question in the chat, Maaike. The question is, what is the advantage of using video instead of a photo?
Oh, because of the video, not all the time when you make a picture, you get a sharp picture of it. Because of the microscope and the mobile phone, it's changed all the time, the sharpness. So when you make a movie, you can let it slow and you can put out the pictures. So that's the reason I make a video of the microscopic what I have.
Other question, Maaike, did you have to reassure your students somehow, and how did you do that? Were they concerned?
What is reassure, Doreen?
Reassure, make them take the concern away. Say everything is okay.
Dat ze een beetje vol zelfvertrouwen aan de slag gaan.
Because I was the first time I was doing it together with them, and I tell them how important this project, and in our class students can choose the subjects. So all the students that choose for this research question about microplastic, they want to do this research, and they were very dedicated to do it on the right way.
And Rob had another question. Do biotic particles tend to move more than plastics? Rob, I'm afraid that most things are no longer living by the time they're under the microscope. So it can be a little bit hard if you prefer to see biology alive, that we tend to not see much live biology by the time we get to the microscope.
I see, sometimes I see amoeba, but that's it. I've looked for the bear, but I don't find it.
What it is?
Yeah.
Okay.
Because you are 10 centimeters below the surface. So because you are 10 centimeter below, there is not a lot of small living particles.
Another question, I think maybe Maaike, you can help. Suppose the children already know how to work with a microscope and you already have the samples. Do you think it's possible to do it in an afternoon? So I think you have the water samples, so do the filtration and the sampling in one afternoon.
In a half hour? 30 minutes?
An afternoon, so I don't know how.
Yes, it's possible, of course.
Yeah.
Yes, I think for watching 500 milliliters, it's take me when you want to see every millimeter, it takes me 2 hours, I think, to see every part. But most of the time, the students make a square of 10 parts that they want to see, and they couch them.
Okay. Thanks so much. I think this is enough, Maaike. Thank you so much for joining us during your class.
You're welcome.
Thank you, Maaike. Great to see you.
Yes. And are there questions still? Please let me know. You can email me and then I can help you.
Yeah, okay. Then you can send me the email. I wrote it in a chat, or it's online, and then I will get you into contact with Maaike.
Yes. And when you're a neighbor, you can maybe we can do it together.
Ah, yeah.
I don't travel to the south, but in the middle, it's okay to help you.
Wow, that's really nice. Okay. Hey,
Okay.
thank you so much.
Thank you. Okay, bye. I go back to my class.
Thank you, Michael.
Thank you, Maaike.
You're
Thank you.
welcome. Bye
Take
bye.
care.
See you soon. Bye bye.
Thank you.
So nothing now. We're
Unfortunately, we cannot offer to travel quite as much as Maaike, but
Yeah.
we can meet you